Welcome to the sequence analysis course. So in this course, we first are going to recapitulate a little bit the WIRE 27 paper, which is supposed to be the paper that accompanies this training, and hopefully you find all these things we are covering here in parallel in that paper where it's being used in a real project. Then we talk about the IGV genome browser. IGV is the Integrative Genomics Viewer, which is a really important tool you always need to use. We will talk about BLAST, the basic local alignment search tool. We talk about annotation formats and then go into sequence mapping and finally SNP calling. All right, quickly recapitulate about the Cariha genome. So this is a wheat genome that was published along with this paper that you can see here. That work has been done in a collaboration between Simon Kratinger's group at the KAUST in Saudi Arabia and Wendy Prince group in South Africa. So Cariha, this accession, is a wheat accession that is being grown in South Africa. So we're talking about Triticum aestivum, the bread wheat genome. And I think at this point, it makes sense to stop this course if you haven't done so already. Have a look at this publication and read through it again, because the idea is that we come up in this course with a little bit of a smaller made up project, but there will be a lot of parallels to this actual project that has been done there. And the idea is that you will find the concepts that we teach here back in the big paper. So please have a read through that again. So let's start with the actual course. And before we do anything about the sequence analysis, I would like to introduce this Integrative Genomics Viewer, IGV, which is a lovely little tool. It doesn't necessarily matter if it's exactly this tool, but what's really important is that we use some tool that we can where we can look into the data and intermediate files we have generated and actually see what was going on. The nice thing about IGV is that we can just install it locally on our computers, so we're not dependent on any servers or any data that is being uploaded by someone else. We just run it ourselves and upload the data we want to look at. So here we first obviously can load genome sequences, and then on top of that we can load annotations or mapping data or SNP data, and we will go into detail what all of this is. So I guess the first exercise is that we go into IGV. We start, we either install IGV on our computers or use the virtual machine that has been introduced in the introductory session. So IGV is installed on the VM. And if you don't want to play with bigger data on your computer, you can use the VM. We'll do both together now. So try to run IGV, try to load the Cariha genome, and then try to load also the annotation. The files for that are linked on our web page, and the data in a very slightly smaller version is also available on our VM. So give it a go yourself. So that means stop this video, pause it, run IGV, and then come back into that, and we'll do it together. Right, so let's have a look together. If you were able to install IGV on your computer, just do the normal steps, and maybe if you have a big enough computer, you will be able to load the entire Cariha genome and the entire gene annotation into your IGV. I'm here in the virtual machine, so this has a little bit less resources right now, and I'm only loading a little part, but the general concept will be the same. So here in the VM, I go back to my terminal, and if I just type IGV, hit enter, it should start up. All right, here we have an IGV. So the first thing we want to do here is we want to load a reference genome. So we click on genomes, click on load genome from file. And then, well, if you're on your own IGV, you would just find the genome sequence, which is in FASTA format. And here we go into our data, references, and we load this locus.fasta, which is just a little subset. It's just one little place in the Carica genome that we can load. Okay, let's open that and have a look at the sequence. You can already see we have about 500,000 base pairs here. So this tells us what we're looking at. If you loaded the entire genome, you will see full chromosomes here. Here you can select chromosomes. Since we have only one locus here, we have only this little piece on chromosome 2b. You might be able to select from all your 21 chromosomes if you loaded the entire genome. So here we can then zoom in a little bit further. And I do that just by clicking on a start place, hold the left mouse button, and then just extend to the part I want to see. And if I zoom in more and more and more, at one point you down here see the actual basis of the genome. So this is not very interesting yet, but it will become far more interesting once we load actual annotation. But what you will already see is as soon as we do something here with the zooming, you will see the positions of where we have zoomed in coming up in this little window. So if you have any region of interest that you want to keep for your lab book or other things, you can just copy this part and paste it somewhere else. So once we have this genome loaded, we can load additional tracks. And at this point, we only want to load the genome annotation. And you can, if you have the entire genome, again, you can load the genome that the gene annotation that was published with a paper. And we have just this little subset that is relevant for our locus. So once we've loaded that, let me just zoom out completely again. You can see there are a couple of blue parts coming up here. And here, now you can see why it makes sense to zoom in like this from one place to a certain different position. We can zoom in on this one genome, on this one gene, and then you can see the structure of the gene. If we right click here on the side, we can change from a collapsed view to an expanded view, which means we see a little bit more. This is the actual locus of the gene. And here we see the intron exon structure and nice pop-up windows, which I turn off now. Never show details for now. So just to give a bit more details on a gene, something like that, we here have these exons, these very small arrows here in the middle are the introns, and then we come to a next exon and have some weird artifacts coming up. Let's zoom out briefly. You might see that in this exon, we have a fat part and a thinner part. So this thinner part here is the untranslated region, the UTR. Okay, but this allows you now to also go more into detail, zoom in. And if you zoom into a certain point, you can see the amino acids coming up and you can see the bases. Okay, so the file that we loaded here, the genome file was obviously in FASTA format. The annotation file was in GFF format. So let's have a look at that file that we loaded here. I just close IGV for now. and go into our Explorer, have a look what we loaded. It was here in data, references, and I loaded this file GFF3 format. Now let's have a look at this file in a text editor. So as you can see, this is almost like a table. Well, in fact, it is a table. It is tab separated format, TSV. And we have at the beginning a little bit of a comment. So you will find in a lot of file formats, if they start with these hash symbols, this is always a comment. So, and everything else here is essentially just a table. We have in the first column the chromosome, and then some additional information. And then we see what kind of feature we are actually looking at, and then a start and an end, a dot, and so on. Let's go a little bit into detail what we have here. So going back to my presentation. What you saw here is something like that. It's a screenshot of it. Let's go back to the description of the actual columns. So the first column was the sequence name. So we can see this is something like chromosome 1A or chromosome 2B. The second part is the source. Where does this come from? And here it just says this is the Cariha version 1 gene annotation. Third column is the feature, and this tells us what part is shown here. And you might see that we have a lot of different things here, and this is actually something hierarchical. So we have a gene that goes from one start to one end. And then we have an ID for that comes in the last info column. But we have also additional features. You can see this has the very same start and stop. So here, for every gene, we might have transcripts. And for transcripts, we might have exons. And for exons, we might have coding sequences, CDS. So the CDS is a little bit of a special case from an exon, because the exons will include the untranslated region, whereas the CDS only goes from the start codon to the stop codon. So I already said the next columns are start and end. And then we have a score, which is sometimes used, sometimes not. When it's not used, we just have a point here. This is a little bit pushed over, because so this column is actually the end, and here's our score. After the score, we have a strand, which is plus or minus strand. This is just on which DNA strand it is, or if we go from the IGV perspective, if it's starting from left and ending to the right, or vice versa. And then we have a frame. So for a protein translation, it can start by the first nucleotide, by the second, or by the third. And the phase or the strand is being displayed here, which is 0, 1, or 2. So if it starts on the first nucleotide in a sequence, there would be a 0 here, and so on. The final column is then the attribute, which is a semicolon separated list of tag value pairs. So semicolon separated, and then tag value pairs. Here we have the tag ID, and then it's separated by an equals, and then this is the actual value. And a semicolon and the next tag value pair. So the ID of this gene is this name. And then we have a gene name, and we have some additional description here. What's more interesting in other features, we have something like a parent. So I said this is a hierarchical thing. So we have a gene ID here, and this ID is referenced as a parent in this mRNA. So you can see this is the very same thing as this. Okay, so I hope this makes sense. This is quite a common format, and you'll always want to go into those text files and make sure everything is correct.